Bismillahirrahmanirrahim. Today, inshallah, we shall continue with our exploration of mitosis and meiosis as cellular division processes. If you recall, mitosis is cellular division of a diploid cell, which results in the production of two daughter cells, which are identical to the parent cell, and they're also diploid. So in mitosis, a diploid cell replicates itself and gives two diploid cells like this. So this is the process of mitosis. Now meiosis, on the other hand, is a very special process where a parent cell, which is diploid, a parent cell that is diploid, and let's call that diploid parent cells, will go through a series of two cellular divisions, first like this, and then these two will go further cellular division processes like this, and the resultant would be actually four daughter cells like this. Let's give myself a little bit of room here, and just to emphasize, these four daughter cells are actually going to be they're completely different in terms of their genetic material to the original parent cell. So let's try to emphasize that, and so this would be, and they all would be haploid cells. So this would be N, 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 and N. So none of them are identical to each other, okay, in terms of their genetic material. So meiosis is actually a series of two cellular division, which takes a diploid cell and produces four haploid gametes. Okay. So before we go on, let's review mitosis as a process of cellular division. So as you recall, in interphase, the chromosomes are duplicated, and by the time prophase ends, the chromosomes have condensed, and the nuclear envelope has disintegrated, and the centrioles, which are these starry things over here, which are responsible for creating the web that are responsible for the dance of the chromosomes, so to speak. And there are two of them over here. Centrioles are moving towards opposite poles of the cell. And here, this is a simple cell with two pairs of chromosomes. So I just want to bring to your attention the two pairs that are in the middle, the small ones, are homologous pairs, and they have duplicated themselves. In other words, one of them is from mom, and that one's duplicated itself, and the other one's from dad of the same chromosome. So they might be chromosome number 3 or something like that. And they're if this was a human cell, and there would be 23, this would be number 3, the pair, and that's duplicated itself. Okay. So and these are also homologous chromosomes, the big ones, and they have also duplicated themselves. Okay, so in this simplified example, what we have here is we're just analyzing what happens to two pairs of chromosomes. Okay, two pairs of homologous chromosomes. They have duplicated themselves, and the centrioles are moving towards the opposite poles and creating the spindle fiber around which the chromosomes are going to line up. And by metaphase, they have lined up and into this line. And this, in fact, is given a name that's called the Metaphyseal Plate. The Metaphyseal Plate. Okay, so in it, they kind of lined up in that plane. Now, again, I wanted to emphasize that there are two pairs of chromosomes. Okay. And this one, for example, is the chromosome number 3 from dad, and this one is chromosome number 3 from mom. They're homologous pairs that have duplicated themselves in this process, which is going to duplicate the whole cell. So, and then these are the centrioles, which have created the spindle fiber, which are now going to separate these chromosomes. Okay, so prophase and then the metaphase. In metaphase, they are lined up in the middle, like this. And then in the anaphase, the individual chromosomes move apart like this, and the spindles fibers pull them into the opposite poles like this. So in metaphase, they meet in the middle, they're lined up in the middle. In anaphase, they start to move away from each other. And finally, in the telophase, the two cells are formed by pinching of the cellular membrane like this, and the nuclear membrane is regenerated, and now you have basically two cells that are, when this process is complete, that is, then you have two cells which are also diploid cells, just like the parent cell was. Okay, so you had started off with a diploid cell which doubled its number of chromosomes by duplicating them, and all the chromosomes lined up, and two cells were formed. Okay, this process, if you recall, if you wanted, like in mnemonic, was summarized by PMAT: prophase, metaphase, anaphase, and telophase, and those are the four phases of mitosis. Now, again, this results in two daughter cells which are identical to the parent cell. Now we're going to compare this process to meiosis, which kind of from a distance looks very similar, but it is quite different in details. And if you appreciate the subtleties, I think it's more enjoyable. So let's try to do that. Now, compare this process to meiosis. Now, meiosis starts out with the same type of cell, and you notice it's a diploid cell. So meiosis starts out with, let's say this would be a diploid cell, and then all the chromosomes are going to be duplicated. And in prophase, just like in mitosis prophase, the nuclear membrane degenerates, the centrioles start to move at opposite poles, like over here, and they begin to line up as they do in the metaphase for mitosis. But meiosis is critically different, is different from the beginning, so to speak, because notice when something exceptionally different that happens. Okay, in mitosis, all the chromosomes line up in the middle like this in metaphase, right, and one chromosome pair, right. But in meiosis, you observe there are two chromosomes pair lining up like that. Okay, now this type of lining up at the metaphyseal plate, this type of lining up where you have what this is called a tetrad, right? Tetrad. This does not occur in mitosis. Okay, if this was to occur in mitosis, this picture here would have to look like, this guy would have to be right on top of the other guy over there, and that's not the case. In mitosis, the tetrad does not form. In meiosis, it's the homologous pairs. Okay, the homologous pairs. That is this. This is dad's, for example, and that's from mom, right? And they have duplicated themselves. So there's two of dads and two of moms because they've duplicated themselves, and they kind of line up at the metaphyseal plate. And in mitosis, the homologous pairs don't line up right next to each other. They are separate, okay? Because one from dad is going to go this way, one from dad is going this way, and one from mom is going to go this way, one from mom is going to go this way, so that both cells have one from dad and one from mom. But now look what happens here. This is dad's chromosome, which has duplicated itself, right? Now both of them are going to go this way. All right, and look at the maternal chromosome. In this case, I'm assuming that's the maternal, which is duplicated itself. It's going to go this way. So the mom's and dad's chromosomes, homologous chromosomes, are going to be separated. So this is a very profound difference between these two processes right in the beginning of the cellular division process, right? So this tetrad that forms, this chromosomal tetrad forms, this does not occur in mitosis. Now, Now, another interesting that happens at this stage of meiosis is, which is not shown in this particular cartoon, is the following phenomenon, okay? And we shall study this again in the future. Now, if you were to magnify one of these tetrads, okay, these are homologous chromosomes, this is from dad to dad and mom, for example, then what can happen is what's called crossing over, okay? So this process is called crossing over. Let me tell you. So this is dad's chromosome, which is duplicated dad, and this is mom's chromosome, which is duplicated itself, mom. So what can happen in this process, okay, is literally they can exchange genetic material. So this part right here actually belong to the dad part of the chromosome, and this, since I used yellow for dad, I will use yellow for over here. So let's say this is dad's chromosome, and then dad will just donate the chromosome to mom, and the mom's chromosome, which is over here, will become part of the dad's chromosome. They'll exchange genetic material. So in the end, this will become something like this. So a set of a new genetic material will be formed like this, okay, as a result of this chromosomal exchange, okay. So mom will have this, like that, okay? So they literally, like, the chromosomes will cut here, and flip-flop. And this allows for genetic variability. So this important process of crossing over of genetic material does not occur in mitosis, okay? But does occur in meiosis. So in metaphase 1, the formation of this tetrad allows for crossing over of chromosome. Okay, so crossing over or sharing of chromosome, which results to further diversity in the gene pool, in the genetic makeup of these cells that are going to be formed. Now, thereafter, just like in mitosis, the cells divide, the spindle fibers pull the chromosomes into the opposite poles, and then that's in anaphase, they move apart from each other, and in telophase, the two cells divide and two daughter cells are produced. Now, there's a difference again that I hope you appreciate, and that is, in these two cells are not the same, because here, for example, this was like the dad's chromosome, which was duplicated itself, has gone to this side, and that's dad. And then the mom's homologous chromosome, the one that corresponded to this son, and it went to this daughter cell. So these two daughter cells are not the same daughter cells. They're actually genetically different, okay? They have 23 chromosomes, but 2 of them are duplicates, right? So that's why, as you can see over here, I didn't write N and N over here and like that, because they're not even same cells, okay? So they're not identical cells. So this is a very critical difference, the formation of the tetrad, which allows crossing over and exchange of genetic material to occur, and the tetrad also causes separation of the two homologous chromosomes. So the homologous chromosomes are now in two different cells. Okay, so now as you can see, meiosis has two parts. The first part is prophase 1, metaphase 1, anaphase 1, and telophase 1. So meiosis 1 has two parts. Meiosis has two parts: meiosis 1 and meiosis 2. So this is meiosis 1, and each of these two daughter cells are going to go on and to give two other daughter cells like this, okay? And then that's, in the end, will form those 4 daughter cells as we spoke about before. So by the end of meiosis 1, the first part of meiosis, this results in production of 2 daughter cells. Okay, so from now on, these 2 daughter cells can live a happy life for a long time. They don't have to do anything. But when it's time, they go to the second part of meiosis. So they can stay like this for a very long time if they wanted to, and but if they want to divide and further, they would go through the same process again: prophase, metaphase, anaphase, and telophase. And but they're called prophase 2, metaphase 2, anaphase 2, and telophase 2, processes that are very analogous to mitosis. Now look what happens now. Now, as we have already suggested, these two are not the same cells because the genetic material has already been separated from one another. So after, if the cell wanted to do division again, you would have to condense the chromatin into chromosomes again, and then in prophase 1 the nuclear membrane would break and the centrioles would form, and the centrioles would move to the opposite end, and in the metaphase the chromosomes would line up in the middle in the metaphyseal plate, and just like in mitosis, the individual chromosome or sister chromatids, they would move to the opposite sides, and that would occur in the anaphase. And then in telophase, the cytokinesis occurs, the cell divides, and you produce a total of 4 cells. Okay, now in this case, then these are daughter cells, and they have, as you can see, 1 chromosome and 2 chromosomes over here, right? And whereas the parent the parent cell had 4 chromosomes, so from dad, from mom, from dad, from mom, right? So this is a haploid cell. So all of these are haploid cells. It's a haploid, haploid, and haploid, and haploid, and these are all different because, as we said, genetic material has been separated. And so this is a brief overview of the process of meiosis, and it's divided into 2 parts: meiosis 1 and meiosis 2. And the key distinction actually occurs in meiosis 1, where in the metaphyseal plate a tetrad is formed and genetic exchange of materials occurs, and then the daughter cells are different. And in meiosis 2 is very similar to mitosis in that in prophase, again, the chromosomes are formed, nuclear membrane disappears, centrioles move to the opposite poles, and in metaphase the chromosomes line up at the metaphyseal plate. In anaphase they separate, just like over here, and telophase of daughter cells are formed. And the end result, if you observe, is creation of 4 daughter cells, which are haploid, as we have suggested over here before. Okay, there are many other nuances to this, and then further details that we can elaborate this on, and we shall do that next time and embellish our knowledge in this way. Until then, as-salatu was-salamu ala rasulillah, assalamu alaikum.